Okay, so we are going to the pistil, that is megasporangium, that is ovule and embryo sac. The pistil megasporangium is the ovule, that is stigma style, and then ovary. So inside the ovary it is an ovule. The arrangement of ovules inside the ovary, that is a platen cushion like structure, that is the placenta, and then ovule. So the pistil, it is a kind of carpel, right? And megasporangium set, that is the ovule and embryo sac. So inside the ovule there is an embryo sac. So this is how, like the pistil megasporangium, that is ovule. Now microsporangium. So what about the macrosporangium? What about the macrosporangium? What about the macrosporangium? What about the macrosporangium? So macrosporangium, it is called like so macrosporangium is equivalent to like a pollen grain, right? The gynoecium represents the female reproductive part of the flower. The gynoecium may be consist of a single pistil, that is monocarpellary, and may have more than one pistil, that is multicarpellary, right? So when there are more than one, pistils, right, may be fused together, that is syncarpus, or may be free, that is apocarpus. So each pistil has three parts, that is stigma, style, and ovary. The stigma serves as a landing platform for pollen grains. The style is the longest slender part beneath the stigma. The basal bulge part of the pistil is ovary. Okay, inside the ovary is the ovarian cavity, that is locule. So inside the ovary there is an ovarian cavity, that is the locule. The placenta is located inside the ovarian cavity. Okay, now there are different types of placentation and all that, like you can see this diagram, it is stigma style, and then ovary, and on the bottom there is a thalamus. So it is stigma. So when it is fused, you can see in the diagram, that is B diagram, that multicarpellary syncarpus, that is fused carpels, pistil of papaver. So it is an example of papaver. So now see diagram, multicarpellary apocarpus, gynoecium of michelia. So it is the michelia, right? So michelia. So it is a michelia, right? Then a diagrammatic view of a typical and anatropous ovule. So you see a multicarpellary apocarpus, gynoecium. Apocarpus, it is free like gynoecium of michelia. So it is the carpels, and then there is stigma, syncarpus ovary, that is papaver, right? And multicarpus apocarpus, that is michelia, right? So it is so anatropous ovule we are going to study, like you can see. So let me first complete the text. So arising from the placenta are the megasporangia, commonly called the ovules. A number of ovules in an ovary may be one, that is wheat, paddy, and mango, to many, that is papaya, watermelon, and orchids. So arising from the placenta are the megasporangium, called the ovules. So these are the ovules, right? Megasporangium. The number of ovules in an ovary may be one, that is wheat, paddy, and mango, and many, that is papaya, watermelon, and orchids. So megasporangium, that is ovule. So with the structure of a typical angiosperm ovule, the ovule is a small structure attached to the placenta by means of a stalk, that is funicle. So the ovule is attached to the placenta with a stalk like the with a stalk, that is funicle. The body of the ovule fuses with the funicle in the region. Now hilum is the region where the body of the funicle and the ovule fuses, that is the hilum. So thus hilum represents the junction between the ovule and the funicle. So ovule and the funicle, the the body of fuses with the funicle in the region called, thus the hilum represents the junction between the ovule and the funicle. So each ovule has one or two protective envelopes called the integuments. The integuments encircle the nucellus except at the tip where a small opening called the macropyle is organized. So opposite the macropyle end is the chalazus representing the basal part of the ovule. So opposite to the macropyle end is the chalazus representing the basal part of the ovule. Okay. So enclosed within the integuments is a mass of cells called the nucellus. The cells of the nucellus have abundant reserve food material. Located in the nucellus is the embryo sac. So located in the nucellus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. An ovule generally has a single embryo sac formed from a megaspore. Now megasporogenesis, just like the microsporogenesis, the process of formation of megaspores from a megaspore mother cell is called the megasporogenesis. Now Ovule generally differentiate a single mother cells, that is MMC, in the micropial region of the nucleus, right? It is large cell containing dense cytoplasm and a prominent nucleus. The MMC, that is megaspore mother cells, undergo meiotic division. What is the importance of MMC undergoing meiosis? Megaspore mother going, the meiosis results in the production of 4 megaspore, now female gametophyte. So now we are going to study the female gametophyte. So in the majority of the flowering plants, one of the megaspores is functional, right? So in a majority of flowering plants, one of the megaspores is functional. While the other three degenerate. So only the functional megaspores. So only the functional megaspore develops into the female gametophyte, that is embryo sac. So only the functional megaspore develops into the female gametophyte, that is embryo sac. So this method of embryo sac formation from a single megaspore is termed as the monosporic development. So this method of embryo sac formation from a single megaspore is termed as the megaspore development. Now, what will be the ploidy of the cells of the nucellus, that is MMC, megaspore mother cell, the functional megaspore, and the female gamete? So what will be the ploidy of the cells of the nucellus? It is diploid. Megaspore mother cell, it is diploid. The functional megaspore, it is, I think, haploid, and female gametophyte, it is haploid. So this is it. Like now, so these are the structures. So now you can see it is like a funicular stalk of the ovule, and it is a stalk of the ovule, and the ovule and the funicle, it is attached and fused at the junction called the hilum. So there is like the micropial end, there is micropial pole, there is outer integument, there is inner integument, and there is nucellus inside, and then there is a embryo sac. Opposite to the micropial pole, it is a chalazal pole, right? So it is a typical anatropous ovule. So it is typical anatropous ovule. So it is typical anatropous ovule, right? Now the parts of the ovule showing a large megaspore mother cells, a dyad and a tetrad of megaspores, that is 2, 4, 8 nucleate stages of embryo sacs and a mature embryo sacs. A diagrammatic represents of the mature embryo sacs, right? So I'm going to convert first text, right? So okay, so the process of formation of megaspores from the megaspore mother cell is called the megasporogenesis. Ovules generally differentiate a single megaspore mother cell, MMC, in the micropial region. You can see it is the micropial end. It has a single nucleus cells. It is a megaspore mother cell that undergoing to the meiotic division. You can see micropial end, then there is a nucellus. So it is megaspore dyad. So it is micropial end. So first are dyad, then tetrad, and then you can see all these things like. So parts of the of the nucellus, right? So in the micropial region of the nucellus, it is a large cell containing dense cytoplasm and a prominent nucleus. The megaspore mother cell undergoes meiotic division. What is the importance of the megaspore mother cell undergoing meiosis? Meiosis results in the production of 4 megaspores, right? So I think I already have done it, right? Okay, let us the formation of the embryo sacs in detail, right? The nucleus, right? The nucleus of the functional megaspores divides mitotically from 2 nuclei, which move to the opposite poles, forming the 2 nucleate embryo sacs. 2 more sequential mitotic nucleate divisions results in the formation of 4 nucleate and a later 8 nucleate stages of the embryo sacs, right? So 2 nucleate, then 4 nucleate, then 8 nucleate, right? So these meiotic divisions are strictly free nuclear, that is nuclear divisions are not followed immediately by the cell wall formation, right? So after the 8 nuclear stage, the cell walls are laid down, leading to the organization of the typical female embryo, typical female gametophyte or embryo sex. Observe the distribution of cells inside the embryo sex. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. So what is that like? So 6 of the 8 nuclei are surrounded by the cell walls, right? So 6 of the 8 nuclei are surrounded by the cell walls, and so 6 of the 8 nuclei are surrounded by the cell walls and organized into cells. The remaining two nuclei called polar nuclei are situated below the egg apparatus. Below the egg apparatus in the large central cell. Okay, there is a characteristic distribution of the cells within the embryo sac. Okay, so there is a characteristic distribution of the cells within the embryo sacs. So 3 cells are grouped together at the macropyle end and constitute the egg apparatus. So 3 cells are grouped together at the macropyle end and constitute the egg apparatus. Now the egg apparatus in turn consists of 2 synergies and 1 egg cell. The synergies have cellular thickenings at the macropyle tip called the filiform apparatus, which play an important role guiding the pollen tips into the synergy. 3 cells are at the chalazal end and are called the antipodal cells. So 3 cells are the 3 cells are at the chalazal end and are called the antipodals, right? The central cell, as mentioned earlier, has 2 polar okay. So guiding the pollen tip into the synergy. So 3 cells are at the like what, where the chalazal end and are called the antipodal cells, right? The last central cell I mentioned earlier has 2 polar nuclei. Thus a typical angiosperm embryo sac and maturity through 8 nucleate and 7 cells, right? So the pollination. So the male and female gametes in the flowering plants are produced in the pollen grain and embryo sacs. So the male and female gametes are produced in the pollen grain and embryo sacs. Right? Embryo sacs is present in the ovule. It is a megasporangium, pollen grain in the microsporangium, right? They are produced. So as both the types of gametes are non-motile, they have to be brought together for the fertilization to occur. How is this achieved? Now this is done by the pollination. So pollination is the mechanism to achieve this objective. Transfer of pollen grains shade from the anther to the stigma of a pistil is termed as pollination. So transfer of pollen grains, that is shade from the anther. Shade is the transfer. So shade from the anther to the stigma of the pistil is termed as the pollination. So flowering plants have evolved an amazing array of adaptations to achieve pollination. So they make use of external agents to achieve pollination. Okay. So there are external agents. So kind of pollination, depending on the source of pollination, can be divided into 3 types. So first one is the autogamy. So in this type, pollination is achieved within the same flower. So transfer of pollen grains from the anther to the stigma of the same flower. In a normal flower which opens and exposes the anthers and stigma, complete autogamy is rather rare. So complete autogamy is rather rare because it exposes the anthers and stigma all the time. Okay, no, complete autogamy is rather rare because the normal flowers are opens. Okay, they expose the anther and stigma. So autogamy in such flowers requires synchrony in pollen release and the stigma receptivity. So autogamy in such flowers requires synchrony in pollen release and stigma receptivity. So autogamy in such flowers requires synchrony in pollen release and stigma receptivity. Also, the anthers and the stigma should lie close to each other so that the cell pollination can occur. Now some plants such as viola, common pansy, oxalis and camelina produce 2 types of flowers, that is chasmogamous flowers which are similar to flowers of other species which expose. These are the like normal flowers with the exposed anthers and stigma, and celastogamous flowers which do not open at all. So in such flowers, anther and stigma lie close to each other. So they lie close to each other. When the anthers descends in floral buds, In flower buds, the pollen grains come in contact with the stigma to effective pollination. So thus cilistogamous flowers are invariably, so they are compulsorily autogamous, as there is no chance of cross pollination on the landing stigma. So cilistogamous flowers produce an assured seed set even in the absence of pollinators. So do you think that cilistogamy is advantageous or disadvantageous to the plant, and why? It is advantageous for the cross pollination for to bring the variation, but it is a disadvantage because it is like continue their inbreeding and all that. So inbreeding and all that things happens in the like chasmogama, in the cilistogamus, right? So you can see the first diagram is of like the cell pollinated flowers, like it has no colors, not that much colors. But the second flower is known as, you can see it, so you can see it is a cross pollinated flower. So the second flower, it is known as the cross pollinated flowers, right? So the second flower, it is known as the cross pollinated flowers, and the third flower is, it is known as the cilistogamous flowers, right? So cilistogamous flowers, they are the they are the they have the floral buds, so they are the closed flowers, right? So chasmogamous flowers, so these are the open flowers. So this is it, like the parts of the wheel showing a large megaspore mother cells, a dyad, and a tetrad of megaspores, and 2, 4, and 8 nuclear stages of the embryo sex and a mature embryo sex, right? Now a C represents a diagrammatic representation of the mature embryo sex, right? So this is like 2 cell stage, 2 nucleated, then 8, and then 4 nucleated, then 8 nucleated, then synergids and eggs, central cell, that is 2 polar nuclei and antipodals, chalazal end, that is the antipodals. In central there is polar nuclei in the central cell, and then there is egg, and then there are like two synergids and filiform apparatus, the micropial end and chalazal end. त्याचा विचार कर ना, तिथे मला नको सांगू तुझी. So this is it, like, so agents of pollination. So agents of pollination, plants use two abiotic, that is wind and water, and one that is biotic, that is animals agent, to achieve the pollination. So majority of plants use biotic agents for pollination, but majority use the biotic agents for pollination, that is the animals, and only a small portion of plants use the abiotic agents. So only a small portion of plants use the abiotic agents, that includes like the wind, you can see wind and water. So pollen grains coming in contact with the stigma is a chance factor in both wind and water pollination. So pollen grains coming in contact with the stigma is a chance factor in both wind and water pollination. To compensate for the uncertainties and associated loss of pollen grains, the flowers produce enormous amount of pollen when compared to the number of wheels available for pollination. Okay, to compensate for these uncertainties and associated loss of pollen grains, the flowers produce enormous amount of pollen when compared to the number of wheels available for the, so they produce in excess. So even if there is loss, it is assured that like is going to be pollinated. Okay, so this is all that, like, so now